So this webinar is more like a technical tutorial of how to perform ChIP-seq data analysis, especially for wet lab biologists that do not have much programming and learning experience. It is also useful for bioinformatics beginners who need to perform ChIP-seq data analysis in future. In this webinar, the starting point is that you've got your raw sequencing read files, most likely FastQ files, and we will only cover sequencing data from Illumina platform. I will show you how to align or map the reads to a reference genome, how to perform peak calling to identify enriched region for the protein of interest, and how to find DNA motifs enriched within the binding regions, and how to assign binding sites to genes and get enriched gene ontology terms to discover potential biological functions. After this webinar, the audience should be able to get a general idea about how ChIP-seq datasets look like and what you can expect from ChIP-seq data. Let me give you an outline about this webinar first. We will cover all the basic ChIP-seq routines by using the software listed in this slide. All the software here, they either have a web-based interface like Galaxy and Great, or they have a graphic user interface like FastQ and seqMINER. So no command line experience is needed here. The datasets I'm using here are FOXA1 ChIP-seq experiments. FOXA1 is a focal transcription factor, and it is one of the first transcription factors that have been chipped genome-wide. We will do a side-by-side comparison of a successful FOXA1 ChIP-seq experiment and a failed one. The successful FOXA1 ChIP-seq data is from the MCF7 breast cancer cell line, published in Nature Genetics from Jason Carroll's lab, shown on the left-hand side. In this paper, the authors discovered that FOXA1 is absolutely required for estrogen receptors DNA binding and activity in breast cancer cells. The failed one is from the novo colon cancer cell line, published in Cell from UC Typeless lab, shown on the right-hand side. In this paper, the authors found that transcription factor binding tends to occur in a highly clustered manner, and most clusters contain cohesins. They performed hundreds of transcription factor ChIP-seq experiments, and FOXA1 is one of them. However, the FOXA1 ChIP-seq dataset didn't pass the author's QC standards, so they labeled this as a failed experiment. This provides a perfect opportunity for us to get an idea about what a failed experiment looks like, which is sometimes even more important than other things, and thank the authors for making this data publicly available. Now, we will start with QC of your sequencing reads using a software called FastQ. It is developed at the Abraham Institute, and you can download it from the website indicated on the top of the slide. As you can see here, FastQ can be run interactively, so you are able to locate your sequencing read files and load them into FastQ by simply clicking your mouse. Here, we have 4 files, 2 for FOXA1 MCF7 dataset, and the other 2 for FOXA1 novo dataset. For each dataset, we got FOXA1 ChIP sample and a control sample. In the MCF7 dataset, the control sample is the input control, while in the novo dataset, the control is the IgG control. Once you load the files by FastQ, you should see something like this in the middle of the slide. It gives you many QC metrics of your sequencing reads. The usage of FastQ is beyond the scope of this webinar, and we won't cover all the metrics in the program. Here, we will only focus on 3 metrics. The first one is basic statistics, which contains some basic information about your sequencing reads, like read length and number of total reads. Well, perhaps the most important piece of information here is the encoding. It tells you how the quality score of the sequencing reads are encoded. The FOXA1 MCF7 experiment is encoded by Sanger Illumina 1.9, and the FOXA1 novo experiment is by Illumina 1.5. We will need this information later, and I will come back to this later. The second metric is read quality. In this graph, the x-axis shows all the individual bases from the reads. For every base, the y-axis is the box plot of distribution of quality scores of all reads across that particular base. The yellow boxes show from 25th to 75th percentile, and the red line indicates the median, and the blue line indicates the mean of the quality scores. You really want the quality scores well above 20 to be good bases. In the FOXA1 novo data on the right, the quality dropped rapidly at the end of the read. In this case, you might want to trim the last or even 5 bases of your reads before downstream analysis. In this webinar, we won't do this just for simplicity. The third metric we are looking at is the sequence duplication level. It tells you how unique of your reads are within the library, and this is a measurement for library complexity. Due to the random nature of sequencing, what you expect to see is that most sequencing reads only occur once, like the FOXA1 MCF7 data on the left. However, in the FOXA1 novo data on the right, the majority of reads occur twice. They are also allowed to occur 4 times. The overall unique sequencing reads is shown above the graph here. The FOXA1 MCF7 data have more than 70% unique sequences, while the FOXA1 novo data have only around 44% unique sequencing reads. The total number of reads in the FOXA1 novo dataset is more than 48 million. So 44% indicates that there are about 21 million unique reads, which is quite okay actually. Now it is worth mentioning that the FastQ results are only indications of the quality of your sequencing, but they cannot tell you whether your ChIP experiments work or not. That's what we are going to do next. Now, after getting a rough idea about the sequencing quality, we are ready to align the reads to the reference genome. To do this, we will use bowtie in the Galaxy platform. Galaxy is a user-friendly online system with many integrated bioinformatics tools. After register and log in to the Galaxy website, which is usegalaxy.org, you need to upload your FastQ files to the Galaxy server. Normally, you just click the Upload file under the Get Data tab on the left. You can choose the file from your computer to upload. However, note here, if the files are too large, they will fail to upload via browser. So it is better to upload your FastQ file via Galaxy FTP. Any FTP client will do the trick. Here I'm using FileZilla as an example. In the host, fill in the address of the Galaxy website, which is usegalaxy.org. The username will be your email address when you registered your account in the Galaxy website, and then fill in the password. Simply click Quick Connect. You will see some log information below, indicating you have successfully connected into the Galaxy FTP. The left-hand side shows the files on your local computer, and the right-hand side shows your files on the Galaxy FTP. They are none at the moment. So now locate your FastQ files on your computer at the left, then simply drag them to the right. The files will start to upload. It is also recommended to zip your FastQ files to save uploading time. The upload speed is of course not as fast as your local network, but it is still quite decent. When it is done, the files uploaded will be shown on the right. Then if you go to the Galaxy website, the files you just uploaded will be shown in the middle. Now we are ready to upload the files from the FTP to the Galaxy server. Simply tick the file you are going to upload. Then, in the File Format drop-down menu, choose the right file format. Our files are FastQ files, and you can see here, there are actually 4 different kinds of FastQ files. The FastQ CS format is for color-space solid sequencing platform, and we won't cover this in this webinar. So the formats we should consider are FastQ Illumina, FastQ Sanger, and FastQ Solixa. The main differences among these 3 FastQ formats are the ways they encode sequencing quality. The FastQ Sanger format uses thread score to present sequencing quality, and it uses the ASCII2 string of thread score plus 33 to encode the quality score. And FastQ files from Illumina pipeline 1.8 and after are encoded in this way. I will show you later what exactly this means. If your sequencing experiments are done recently, so most likely your FastQ files will be in FastQ Sanger format. The FastQ Illumina format uses thread score as well, but it encodes the quality by ASCII2 string of thread score plus 64. FastQ files from Illumina pipeline starting 1.3 and before 1.8 will be in this format. If you remember from the previous FastQ-C result, the FOXA1 novo chip sample is from Illumina pipeline 1.5, so it should be in FastQ Illumina format. And the rest of the 3 samples are all from Illumina pipeline 1.9, so they will be in FastQ Sanger format. The FastQ Solixa uses a different system to encode the quality score, and it is highly unlikely that your data files will be in this format, unless you are analyzing some very old Solixa data. More information about this can be found in this paper from NAR and from the Wikipedia page at the bottom. So now, tick the file and choose the right file format. Click the Execute button. The file will start to upload from the FTP to the Galaxy server. After they are done, they will appear at the right-hand side with green color. If you click the little I button here, you will see what is actually in the file in the middle. We use the FOXA1 MCF7 data as an example. Basically, the FastQ file is just a simple text file. Each sequencing read is represented by 4 lines of text. There are about 27 million reads in this sample, so there are about 108 million lines of text in this file. Let's look into details of one read shown here on the top in the black box. The first line is the name of the read, and it must start with an @ character. The second line is the actual DNA sequence of the read. The third line means nothing, but it must start with the plus character, and only this plus character is required. The rest of the line is optional. Since I get the data from SRA, this line is the same as the first line except the beginning character. And most FastQ files only have a plus character at the third line of each read. In this way, you reduce the file size and save storage space. The fourth line is the ASCII2 string of quality score encoding at each base. Here is an ASCII2 table that you can easily find via the internet. And let's look into each base of this read. So the first base of this read is A, and the quality string for this base is B. By looking at the ASCII2 table, we can find that the ASCII2 code for string B is 66. Since this is FastQ Sanger format, the thread score will be 66 minus 33, which is 33. A thread score of 33 indicates that the error probability of this base is 0.0005. And the second base of this read is G, and the quality string for this base is the @ character. Again, by looking at the ASCII2 table, we found that the ASCII2 code for the @ character is 64. Then the Fred score for this base will be 64 minus 33, which is 31. A Fred score of 31 indicates that the error probability for this base is 0.0008. And you can check every base within this read by yourself in this way. And that is basically how you read and interpret the data from FastQ files. Now, we will use bowtie to align the sequencing reads to the genome. But the bowtie software in Galaxy only supports FastQ Sanger files. So we need to convert the FOXA1 novo chip file from FastQ Illumina to FastQ Sanger. To do this, on the left-hand side, click FastQ Groomer under the NGS-QC and the Manipulation tab. Then in the middle, choose the file you are going to convert and the input format, and hit Execute. After it's done, it will appear on the right with green color. And you can click the pencil button here to rename it as something meaningful like this. Finally, we are ready to align or map the sequencing reads to the reference genome. We will be using the bowtie program. So on the left-hand side, click Map with bowtie for Illumina under the NGS Mapping tab. Then in the middle, select the reference genome. In this case, all samples from both experiments are from human cells. So we choose HG19 here, which is not the latest, but it is a stable version of human genome assembly. For the input file, choose the FastQ file you want to map, and in the bowtie setting, choose full parameter list. Then you will get many options. Now, we will keep everything with default, except change this -m option to 1. So what does this mean? To put it simple, for some reads, they can be mapped to multiple locations within the genome. And you can handle this read differently according to your need. But now, we put 1 here, which simply discards those reads. This is a good starting point for beginners. And actually, many people are still doing this when they analyze DNA-centric sequencing data, like ChIP-seq, DNA-seq, and ATAC-seq. So you do the same for the other 3 files, and after they are finished, they will appear on the right-hand side in green color. As you can see here, I already renamed them to something meaningful. Again, you can click the I button to actually see what's inside in the output alignment file. The output alignment file from bowtie is called SUM file. The SUM file is also a simple text file, with each line representing a read containing the location it is mapped into the genome and other information. The details about the SUM format can be found in the PDF link shown at the top. Now, after mapping, we are going to perform peak calling to identify FOXA1 binding sites. However, the SUM files at the moment contain both mapped and unmapped reads. We only want the mapped reads for the peak calling, so we need to remove all the unmapped reads. To do this, we will use SUM tools shown on the left-hand side. Just click Filter SUM or BAM, then in the middle, choose the file you want to filter, and choose Filter on bitwise flag to Yes. Then you will get more options. In the skip alignment with any of these flag bit set, tick the read is unmapped. So in this way, SUM tools will remove all the unmapped reads. After it is finished, the output will appear on the right in green color. And I already renamed them to something meaningful. By default, the output from SUM tools are BAM files. The BAM file is a binary file, not a text file, so you won't be able to view the content. You can simply think the BIM file is just a compressed SAM file. It significantly reduces the file size. Okay, now we are ready to perform pig calling. For this, we will be using a very popular pig caller called Max, developed in Shirley Liu's lab. On the left-hand side, under the NGS pig calling tab, click Max to use it. In the middle, there are many options, but we will only change a few of them. First, give it a meaningful name. Then, choose the chip file, which is the FOXA1 chip file, and the control file, which is the matching control experiment, input control for MCF7, IgG control for novo cells. The default genome size is 2.7 billion base pair, and this is for human cells. For different organisms, check the Max website for the genome size. Then change the text size to your sequencing read length. Pick the path Excel files into interval files. We will see this later. Then choose Save Weak files so that you will get the binding signal files later for visualization. Then last, choose Do Not Build a Shifting Model and 100 base pair for arbitrary shift size. So why change these two options? As you know, that for a regular ChIP-seq experiment, the sequencing reads you get are from the ends of your fragments, which are not the actual TF binding size. The actual TF binding size are somewhere in the middle of the fragment. By default, Max will estimate the fragment length and shifting the reads to the middle by half of the fragment length to represent the actual TF binding. However, for some reason, sometimes it cannot reliably estimate the fragment size. A common practice is to simply disable this and ask Max to shift reads to a certain distance. In this case, we choose 100 base pair, which is the default when you disable the model build, and it works quite well. Now click Execute. After it is finished, you will get some outputs on the right-hand side in green color. And the number of output files depends on your Max setting. Well, in this case, we have 6 output files for each experiment. Now, let's have a look what each file is. The first file is the HTML report containing the running log of Max. The second and third files are weak files containing the signal intensity across the genome. The treatment weak file is your chip sample signal, and control weak file is your control sample signal. Again, the weak files are simple text files. We won't look at the negative peaks interval files here. The fifth file here is a peak interval file containing the enriched region of your protein, in this case FOXA1. The last file is a bad file for visualizing peak position in a genome browser. And both the interval and bad files are simple text files. If you click the name of the interval file, and you can download this interval file to your local computer, and it can be opened and easily handled and manipulated by any spreadsheet software like Excel. This is probably one of the most important output files. It contains all the information you need to know about the binding sites, including the location and the statistics. Well, at this stage, the p-calling is done. A common and tricky question that most beginners have to face at this stage is that, how can I tell whether my experiment works or not? Actually, there are many things you can check, but the following 3 things are the most efficient way of telling whether the ChIP experiment works or not. First, by looking at the interval files. Second, by visual inspection of the binding signal. And third, by looking at the enriched motif within the binding site. We will now go through this one by one. We will start with examining the interval files. To do this, you sort first by FDR, smallest to largest, then by fold enrichment, largest to smallest. The first thing we should look at is the number of binding sites. If you choose a FDR cutoff at 1%, which is commonly used, there are about 42,000 peaks left in the FOXA1 MCF7 data, but only 430 peaks left in the FOXA1 novo data. Of course, the number of binding sites depends on the protein, the peak color, the cell line, and many other things. But typically, the number you should expect is from around 1,000 to several thousand, or even several tens of thousands. As you can see here, the FOXA1 novo dataset apparently do not satisfy the expectation. The second thing we could look at is the range of the fold enrichment over local background. The FOXA1 MCF7 data have a wide range from more than 300-fold to about 4-fold, while the largest FOXA1 novo peak has only around 20-fold enrichment, indicating the signal-to-noise ratio in this data is really low. The third thing we could look at is the number of sequencing reads within the peak region. Again, the FOXA1 MCF7 data have a much wider range than the FOXA1 novo data. So these three metrics we just went through clearly suggest that there is something wrong with FOXA1 novo dataset. And the next thing we could do to judge the quality of the data is to visualize the binding signal. And we found this seems to be the most efficient way of telling whether a ChIP-seq experiment works or not. So the weak files generated by Max are the signal files, and you can visualize them directly in the genome browser. However, for large datasets, it is highly recommended to convert the weak files to big weak files before visualization. The big weak format is an indexed binary format. When you visualize the big weak files, only the portions of the files being displayed are transferred to the genome browser, which is much faster than loading the weak files. So to convert weak files to big weak, on the left-hand side, click the weak bed graph to big weak under the Convert Format tab. Then in the middle, choose the weak file you are going to convert and click Execute. After it is done, change the file name to something meaningful and click the name of the file. You will see an option of Display at UCSC main. Then click that. A new tab or window of UCSC Genome Browser will appear. In the Genome Browser, to configure the visualization, set the display mode to full so that it will display the histogram of the signal. Set the vertical viewing range from 0 to a reasonable maximum value, like 100 or 200. And set data viewing scaling to use vertical viewing range setting, so that it won't automatically rescale. You would be able to visualize the binding signal like shown here. Now, what kind of patterns are we expecting? There are a few things you can look at this stage. You can have a look whether the peak shape is normal, i.e., whether they are smooth bell curve shaped peaks or some strange spikes. And if you have some non-target genes, can you find some binding peaks in their promoters or near the transcriptional start sites? And finally, to get a bigger view of the binding pattern, you can look at the whole chromosome view. This is an example of signals of all 4 samples across the entire chromosome 12 of the human genome. The 2 control files are almost flat as you would expect them to be. The FOXA1 MCF7 chip sample has many peaks, but the FOXA1 novo chip sample looks very much like its matching control sample. There are only a few very small peaks. Now, this is a clear indication that the FOXA1 novo chip experiment failed, or at least it is not optimal. So that's how you can judge the quality of the data visually. The next step is to identify enriched motifs within the binding sites. We will use a program called MIME-ChIP to do this. In order to use MIME-ChIP, we need to extract the DNA sequence from the peak coordinates to a FASTA file. Now I need to clarify two terms before we go any further. When max is used for peak calling, peak means the whole enriched region from start to end, indicated by the black bar on the top left, while summit, indicated by the thin orange line, means the highest pile-up point within the peak region. It is supposed to be the exact TF binding site. It is not a good idea of putting too many sequences for the motif discovery, and a common practice for TF motif discovery is to use the 100 base pair region centered on the summit as the input for motif discovery. So the column E in the max interval file is the distance of the summit to the start position. So to create the coordinates of peak summit plus minus 50 base pair, just create 4 new columns after the FDR column of the interval file. The first new column is the same as column A, which is the chromosome name. The second column will be column B plus column E minus 50, and the third column will be column B plus column E plus 50. In the fourth column, give a unique name for each individual region like this, and you can do this very easily using Excel. Now, save these 4 new columns as a tab delimited text file. Let's call it FOXA1 summit 100bp.txt. For simplicity, I will only choose the top 1,000 regions for motif discovery in the webinar. Now, we are going to extract the 100 base pair DNA sequence of these regions using the coordinates. To do this, first upload this text file to the Galaxy server using a web browser. On the left-hand side, click Upload Data under the Get Data tab. Then in the middle, choose the file format as interval. Choose the text file you just saved on your computer. And make sure the genome assembly is correct. After uploading, click Extract Genomic DNA under the Fetch Sequences tab. Choose the file you just uploaded and simply click Execute. When it is finished, you will see the DNA sequence within each 100 base pair region in a FASTA format. And you can save this FASTA file to your local computer. Let's call it FOXA1 summit 100bp.fasta. Now, go to the MIME-ChIP website using the address at the top of the slide. The options here are pretty much self-explanatory. In the input, choose the FASTA file you just downloaded. Then enter your email address and give a job name. The default setting is always a good starting point, so you just simply click Start Search. Then you will receive an email with a link to retrieve the results. The result page will look like this. The motif found at the left-hand side is the de novo motif that are enriched within your input region. And the known or similar motif on the right-hand side indicates the similarity between this motif with known TF motif. Now shown on the screen is the FOXA1 MCF7 data. The top motif is a typical 4-cat motif with a very small E value. This is pretty much what you expect from a successful experiment. There are also some other motifs returned, indicating potential interactions with other transcription factors. And if the family motif of the transcription factor you are chipping is not enriched in the binding region, like the FOXA1 novo dataset we are going to show in the next slide, then you need to put a question mark on the data, unless you have other strong evidence suggesting that the experiments are working. Now shown on the screen is the motif results from FOXA1 novo datasets. As you can see here, although the top two motifs look like the 4-cat motif, the E values are very big. There are also two motifs with very low complexities, which are just simple repeats. This motif results further confirm that the FOXA1 novo chip experiment is not working. Now we have finished the motif discovery step, and we had a better impression about the quality of the data. Another thing that most biologists are interested in is to assign the binding sites to genes and perform gene ontology analysis to find potential biological functions of transcription factors. To do this, we'll be using a web tool called GRADE, developed in Gil Bijirano's lab. It is very straightforward to use. Just go to the GRADE website using the address shown on the top right. Then in the genome assembly, choose the red genome, in this case is HG19. Then choose the peak file from your computer as test regions. Since this is a ChIP-seq experiment, choose the whole genome as background. If you click Show Settings, you can see how exactly GRADE assigns the peaks to genes, and you can change it as you want. But according to the paper, the default setting seems to perform the best. So just click Submit. The output looks like this. The top contains some basic information about peak gene association, and the bottom contains the enriched goal terms from specific categories like molecular function, biological process, and the cellular component, as well as a lot of information from many other different databases. It is quite informative to look at those, but we won't go through them here. One thing that is particularly useful is that if you click the job description on the top, this section will expand. And click View All Genomic Region Gene Association. You will see two tables. One gives you for each peak its associated genes, and the other gives you for each gene its associated peaks, together with the distance between the peak and the gene. And you can download them as a text file and save for future use. This is quite convenient. Now we have finished the gene ontology analysis. Last, but not least, it is a good way of showing binding signal in Heatmap. To do this, we will need the alignment files, which are the BAM files after we created after the mapping earlier. So click Download Dataset to save the BAM files to your local computer. We also need a program called seqMINER, developed in Lazaro Torres' lab. Just download seqMINER from the address at the top. Run it according to your operating system. Then you should see an interface like this. First, to load data into it, in the Load Reference section, choose the peak file, for example, the 100 base pair region text file we just created. Since this is a small file, it is immediately loaded, and you can see the information in the upper center. In the Load Aligned Reads section, choose the BAM files just downloaded, and then click Load Files. These two files will be loaded into memory. and this seems to be the time-consuming part of all the analysis in seqMINER. After it is done, the data set will be shown in the middle. When you click Extract Data, seqMINER will plot the reads from each alignment file you just loaded on top of your peak file and generating heatmap matrix. After it is done, the result will show up on the right. If you right-click the result, choose Visualization of Heatmap, a heat map of the two files you loaded will be displayed. You will also have a density plot at the bottom right. The good thing about seqMINER is that you can load multiple experiments at once and easily perform k-means cluster to find different binding patterns and extract different clusters for more detailed analysis. But that is beyond the scope of this webinar. So far, that's all the basic ChIP-seq routines done without any programming and command line involved. Hopefully, by running the workflow in the webinar on your own data by yourself, you would quickly have a general idea about how ChIP-seq datasets look like and what we expect from ChIP-seq dataset. Here, I put some references, mainly from Nature Protocol, containing detailed step-by-step guidance, including troubleshooting tips of the key aspects of ChIP-seq analysis. Some of them require some programming or at least command line experience. Well, it is highly recommended for wet lab biologists who are doing genomic experiments to learn some basics about Linux usage and perform the data analysis yourself. It makes you have a better understanding of the data itself. And once you've done that, all the things we just went through in this webinar can be done in just several simple commands. Okay, this is the end of my presentation. Now I will hand over to Mariam, who will introduce some related products and protocol from Abcam. And then I will come back to answer questions. Thank you.
Thank you, Xi, for such an interesting and comprehensive presentation. And I would like to take this opportunity to talk to you about some Abcam products and resources. We have just launched a high-sensitivity chip kit that has been specifically designed to be used when there is a limited amount of sample available to perform chip. For example, when working with patient material, transgenic mice tissues, or stem cells. With the high-sensitivity chip kit, you can obtain enrichment of your sequence of interest by using as little as 2,000 cells or half milligram of tissue per reaction. You can get your result in only 5 hours, so the experiment can be done in 1 working day, and the eluted DNA can be used straight away in sequencing or microarray or qPCR. Our high-sensitivity chip kit is part of our range of high-sensitivity products designed for when there is a limited amount of starting material. If you want to create a DNA library with limited amount of DNA, our high-sensitivity DNA library preparation kit for Illumina sequencing allows you to create a DNA library from only 0.2 nanograms of DNA. And our ChIP-seq high-sensitivity kit combines the benefit of the two kits I've just mentioned. This kit is designed to carry out a successful ChIP-seq starting directly from as little as 10 to the 5,000 cells. Biosulfite sequencing is the use of biosulfite treatment to determine the pattern of methylation. To help you to take your methylation pattern studies further, we now offer a couple of products to prepare post-biosulfate DNA libraries for Illumina. With our post-biosulfate DNA library preparation kit, you can prepare a library using pre-treated DNA in only 5 hours. If you haven't treated your DNA, then we recommend our Biosulfite Seq high sensitivity, which has all the reagents to perform the biosulfate modification, followed immediately by a DNA library preparation step. The library is modified and ready in only 6 hours. Without delay, I will pass the microphone back to Xi, who is ready to answer some of your questions.
Okay, thank you, Mariam. The first question is that, is paired end or single end read data better? Well, for most of ChIP-seq data, single end data is a single end experiment is more prevalent. If you do pair end sequencing for a ChIP experiment, of course the cost will be much more than the single end data. But if you look at the information you get from pair end data, in terms of ChIP-seq, you don't get a lot compared to the single end data. So in a cost-effective way, most people still do single end data in terms of ChIP-seq. And the second question is that, is input or IgG a better control? Actually, they are both okay for ChIP-seq experiments as controls. Different people use different controls. For us, we prefer the input as a control because it provides much more complex libraries. And the third question is that, is there a simply way to quantify changes in peak height between two ChIP-seq experiments using the same antibody, using chromatin from different cell types or conditions? Well, this is a very tricky and very good question. Now there are, in the ChIP-seq communities, a lot of people have put in a lot of effort to developing the software to identify differentially bounding peaks, but at the moment there's no consensus about which method to use. That there are already a lot of methods out there. If you Google differentially bounding ChIP-seq methods, then I'm sure you will get a lot. And you can have a look the math behind the methods and choose by yourself. And then another question is that, what are the differences between reads and peaks? So the read is the raw sequencing read, that you get from the sequencing machine, containing the actual DNA sequences and the quality scores. Nowadays, they are usually between 50 base pair to 100 base pair. After mapping, the mapped reads contain the genomic locations where the reads are aligned. So the mapped reads will be the genomic coordinates of 50 base pair to 100 base pair long, depending on your sequencing length. Well, peaks are the genomic regions returned by the peak caller. They are the regions where the reads are aggregating or pile up and pass certain statistic threshold of your peak caller. And then this is the last question I will answer. So the question is, if the number of bounding sites falls below 1,000, should I consider my experiment has failed? The short answer to this question is that no, you cannot tell that whether your ChIP-seq experiments fail or not solely based on the number of bounding sites, because the number of bounding sites depend on many things, like the expression of the protein, the cell line, the peak caller, and also the cutoff threshold used in the peak calling. If you use the method in this webinar, the number we normally get from transcription factor ChIP-seq is around 1,000 to several thousand. Well, if the number of bounding sites fall below 1,000, you need to put a question mark. However, you cannot simply judge whether the experiment works or not solely based on the number of bounding sites. You also need to check other things like motif result and visual inspection of your signal intensity. Okay, so this is the last question, and thank you all for attending this webinar.